Good day, everyone. This is Dr. Chris Pryor, and we have a totally unique opportunity here in that this represents the life work of Dr. Kester, who's presenting today. We're working with a family of ceramides that completely block cancer growth. They are the natural mediators, the natural defense mechanisms of blocking cancer. They only target cancer cells. They're in every healthy cell in our body. They are therefore cytotoxic, they're potent anti-cancer agents, and are safe. Very important statement. They work in a very broad range of cancers, and they also have a very unique dual mechanism of action in terms of activating the immune system to attack cancer cells, and specific inhibition, safe inhibition as the natural inhibitors of STAT3 target. Lastly, with the cancer cells trapped in this manner, they become very susceptible to chemotherapy and reactivating chemotherapy, which is terribly important for relapse patients who are no longer responding to frontline therapy and need restoration of activity to prolong their lives without adding any further side effects. Very important unmet need. So these are the fundamentals of why this is a unique opportunity. And on the next slide, leading the charge is the management team. Can I have is the management team led by Dr. Dan Vlok, our Chief Medical Officer, who has tremendous experience at large phase clinical trials in large pharma, Pfizer, Boehringer Ingelheim, J&J. He also brings to the table tremendous commercial experience at developing oncology products. I parallel his background in terms of 30 years plus pharmaceutical development experience at companies like Biogen, Aventis. I've got 4 product approvals, and my last 4 companies in a row, I've had 2 M&As and 2 exits entering the public markets, of which I ran a public company. Basically, the team has done this before. But I'm also proud to announce or include that our scientific founders are very much involved with the company, because we are also a technology platform company that specifically, uniquely delivers these ceramides. We're the only ones that can do this. So we're very excited about that. So in the next slide, so what is our current status? What are our objectives? And what is the fundraise? So you can see the landscape. We completed a phase 1 dose escalation with a particular ceramide that has particularly potent anti-cancer properties. And the design was really intended to demonstrate safety and half-life. But at the expected dose range that we would have predicted from the massive preclinical work, we saw a distinct signal of efficacy. So with that, and based on our KOLs and clinical advisors, our objective then is to move the solid tumor program and the AML program into phase 2A next year. So to do that, I will be taking you through the development timelines, the use of proceeds through to our various milestones. But initially, we're seeking a $10 million raise to milestone 1 and a follow-on of $20 million to milestone 2. And so with that, I'll turn this over to Dr. Kester, who will describe the mechanism of action, the preclinical data, and he'll tag team with Jeff Davidson on the clinical side. Thank you.
Thanks, Chris. I'm Mark Kester. I'm CTO and co-founder of PendreaBio. The beast is on the upper right-hand corner. That's N-hexanoyl deerythrosphingosine. It's a ceramide. It's a pro-apoptotic lipid. Unfortunately, ceramides are immunogenic, and they are precipitating, and they fall out of solution, and you have to come up with a delivery system. We've come up with that delivery system. It's called Ceraxa. That is our IP that differentiates us for everybody who's trying to put ceramide into the body as a pro-apoptotic lipid. The Ceraxa formulation is shown on the left, and bottom line is the Ceraxa formulation increases the half-life in the body in humans is north of 24 hours. And our Ceraxa formulation has a benchtop stability, at least according to the FDA, of 24 months, i.e. it is a product. Next slide. So here's a cartoon showing the two mechanisms of action of Ceraxa. You get two bangs for the same buck. Number one, Ceraxa dephosphorylates STAT3, which inhibits through its post-translational mechanisms, glycolytic pathways, and also pro-survival pathways. In addition, Ceraxa can turn M2 into M1 macrophages. Those macrophages are more inflammatory, and they actually resurrect the immune system, which is downregulated in cancers. So the M1 macrophages activate CD8-dependent T-cell attack upon the tumor, and you get a double whammy of cell death upon the cancer. Next slide. So in a second cartoon format, most chemotherapy, in fact, all chemotherapy, the chemotherapy leads to endogenous ceramide production. But eventually, the ceramide endogenously runs out and the chemotherapy fails. What we are doing is we're adding exogenous ceramide through the Ceraxa formulation. In that way, we can maintain ceramide metabolism, we can also maintain its pro-apoptotic actions, and in reality, we can resurrect, therefore, the ability of chemotherapy to continue to drive the pro-apoptotic effects of that chemotherapy, i.e. exogenous therapy drives endogenous production. Next slide. So here is actually the phase 1 trial results. Notice that most importantly, 5 of the 13 patients ended up with disease-free, stable disease. That is that there were no secondary metastatic lesions and or the tumors that we saw did not increase by 20%. We did not expect that type of efficacy. We were just doing this to show that we were basically non-toxic. We show that upon going up to 323 mg per meter squared, we have no maximum tolerated dose determined. We have no dose-limiting toxicities, and yet we still have effects as a monotherapy. And that's important because we're going to see in the next couple of slides that 1 and 1 do not equal 2. 1 and 1 often equals 11. Ceraxa can actually increase the efficacy of chemotherapeutics. So seeing it as a monotherapy efficacy is really exciting as we move forward. Next slide. So here we're actually looking at some liver cancer data. We have 2 different animal models. On the left, we see a trial where we actually add cancer cells directly to the spleen. They form punctate nuclei within the liver. We then treat with carbon tetrachloride. We end up with a cirrhotic fibrotic liver, and we recapitulated humanized liver cancer. We see Ceraxa compared to Ghost formulation, which is the same nanoliposome, no bioactive ceramide. We actually not only a decrease in growth, we see a regression of a solid tumor. And that's kind of amazing, and that's one of the major things that we are excited about as we go forward now into phase 2 trials. Based upon that study on the left, which is 42 days, we then did a trial on the right where we actually treated only for 42 days. We then went cold turkey, and we took away the Ceraxa, and we saw the animals that got pre-treated with Ceraxa and then went cold turkey, lived a complete lifespan compared with the ghost-treated animals that actually succumbed to the disease around 120, 130 days. Next slide. So we now go from liver, we're going to prostate. Here we're showing that 1 and 1 equals 11. Low-dose Ceraxa, low-dose androgen receptor blockade, abiraterone. We can show that 1 and 1 equals 11, both at the level of viability, cell death, and proliferation. Next slide. We then show another model. Here's pancreatic cancer. All this has been published. We are happy to give you the 20 to 40 papers that we've published on this already. But in this paper with pancreatic cancer, we see that low-dose Ceraxa and gemcitabine additively lead to a further regression in tumor volume in an in vivo orthotopic model of pancreatic cancer. Next slide.
So I'm going to give
this data. I'm going to give it to Chris Davidson.
All right, great. Thanks so much, Mark. This is a schematic showing our design for our solid tumors trial. And so we're looking at treating 4 different kinds of cancer: pancreatic, liver, ovarian, and prostate, and establishing a dose combination regime that works well for those different cancers. That gives us several advantages. One, we're already using the treatments that are in use for those cancers, and we're combining them with Ceraxa to make it even better. Secondarily, by making it better, and treating relapsed refractory patients. These are patients where the traditional chemotherapies have stopped working. And what we're seeking to do is to restore that working, the function of those chemotherapies, and bring about some cell death and tumor control. Based on those dose settings in the phase 2A, our recommended phase 2 dose combination, we're going to proceed to adding additional patients at those doses. That'll give us some evidence about how well those materials work, how well they work in treating that type of cancer. In the phase 2B portion, we're going to expand and treat additional patients in the best responding indication, whether it's pancreatic, liver, ovarian, or prostate. And so that really gives us an opportunity to treat relapsed refractory and to treat them with the current therapies in a very solid way. So we're excited about this trial design. And so I'll turn it back to Mark to talk a little bit more about our acute myeloid leukemia program.
Thanks, Jeff Davidson. So on the left, we're now looking at a model of AML. Here we're actually using Ceraxa to increase the efficacy of the conventional chemotherapy, which is low-dose Ceraxa and venetoclax. And we see with the triple combination, we've extended the lifespan. And this is one of the trials that Jeff's going to be talking about momentarily. But in addition, we're really excited about the data on the right. Ceraxa actually causes a little autophagy. The cancer cells want to survive, so they eat their own organelles for energy. If you inhibit autophagy, and here we're using vinblastine, which is normally not an AML treatment, but together with vinblastine, 67% of all animals led to some sort of disease-free remission. That's kind of exciting and something we are also thinking of going forward to in a phase 2 trial. Next slide, Jeff.
So similar to the solid tumors program, we'll have a phase of monotherapy followed by a phase of dose combination, followed by testing for efficacy. As Mark mentioned, we're excited about this program. We think that Ceraxa plus low-dose Ceraxa and venetoclax could be a very powerful new therapy for relapsed and refractory patients.
So thank you, Jeff. Back to me. So I'm going to take you through the use of proceeds and the value inflection points. So to complete the first part, the very first part of the phase 2A, the recommended phase 2 dose, we require 10 million. We have raised part of that already, but we really, our goal is to raise the 10 million so that the company is in a very strong cash position at that value inflection point 2, value inflection point 1. Then we will do a follow-on of 20 million to get to the end of phase 2A at the value inflection point 2. And just to remind you, these are really high value creating inflection points. We'll have, for example, 4 overall response rates in 4 solid tumors and the AML. If we go into year 3, we will require an extra 20 million to prepare for phase 3 and hopefully accelerated approval. But these value inflection points jump enormously going between 1 and 2 and obviously 3. So what is our confidence to get there? On the next slide, the answer is we are very confident for very fundamental basic reasons. The signal that we see in monotherapy in the phase 1 will be greatly increased in combination because of this synergy. We have multiple indications, multiple shots on goal for solid tumors and the AML. We have a low bar for approval looking at this terrible unmet need, restoring the activity in relapsed patients, but most importantly, well, it's all important, is the safety of this drug as it exists in nature. So thank you very much for listening to this presentation, and we'll be glad to follow up on any inquiries and introduce and bring in our Dr. Dan Vlok, our Chief Medical Officer, to take you through the clinical trial design and the unmet need and why we must get this to patients. Thank you very much indeed.